Hello, my name is Michelle Morand, and I'm here with the cancer guy, Alexander Rolland. Hi, Alex. What's going on with you today?
I just got out of sinus surgery, and it was a
Oh,
fairly intense sinus surgery. They rebuilt my nose, took out a polyp, and put a couple of little tubes in the nose. So I have to wear this strip now to keep my nostrils open so I can breathe properly.
okay. Is this like a lifetime? You'll be like wearing the strip for the rest of your life, or?
Well, I guess based on the fact that I'm aging and I keep walking into doors and so on, I would say that this is probably going to be something you're going to see a lot more of in the future.
Great. Okay. Looking forward. So today, we're here to talk about something that I haven't seen you this jazzed up about, a new type of therapy
Oh,
for.
come on. I get jazzed up about every type of new therapy.
You do.
Remember immune therapy? Remember PD-1 inhibitors a good 8 years
I'm
ago?
just saying, your excitement about the standing ovation at EMSO and various other things,
Yes.
this is big stuff. So I'm going to let you, you've created a little presentation for us about this therapy. I might chime in with a question or two. Tell us a little bit about what we're talking about today and why you're so excited about it. And specifically, how is it going to benefit the cancer patients, and so many of them too?
Right. Well, as you know, precision oncology is defined as the right drug at the right time for the right person.
Mhm.
And so having all three of those conditions met allows us to really focus on what's best for a specific cancer patient and get a lot of efficiency with minimal side effects. And this is just a huge step in that direction. As a scientist, I'm all about molecular mechanisms. Everything in science is about molecular mechanisms. Do you have a defined mechanism to prove the behavior? And really, that's where we start out with in cancer drug discovery. Whereas medicine is more of a statistical approach, where they will take a drug and let's say give it to 5,000 people with a primary tumor in the breast, and then see how many people it works on, and use it in that way with no real understanding of the molecular mechanisms. So, as a precision oncology specialist, I've really focused on mechanisms.
Mhm.
And one of the greatest mechanisms that we have right now is a new discovery, a new class of drugs called antibody-drug conjugates. You've probably heard about them. They're all over the place. There's been many technologies that have really helped with this. Three different technologies that have been independent. Number one is the technology of developing antibodies. We can produce antibodies with just about anything now, and they're really cheap to produce, and they can be done fairly easily. The second thing is the linkers. And these are molecular scaffolds that attach the antibody to a chemotherapy agent. And then, of course, the third level of development is on the individual chemotherapy elements. And so ADCs are basically an antibody against a cell surface receptor or a antigen. And an antigen is something that is on the outside of a cell. It helps the body understand what's in that cell. It's kind of like a street address. But think of these as external proteins that can be utilized in that certain cancers will highly overexpress them. They'll have a lot more of these receptors or proteins on the outside, and we can use these antibody-drug conjugates to target those cancers. And more importantly, with antibody-drug conjugates, they are effective regardless of the type of cancer you have or the primary tumor, because they only care about whether you have that cell surface receptor or not. And if it's not there, then the drug won't bind and it won't release its chemotherapy agent. The second thing about these drugs is when they're developing chemotherapy agents many, many years ago, and I don't think we've developed a new one for quite some time, over 20 years. I think TAS 102 is probably the last concoction. Over time, they've developed really strong chemotherapy drugs. And these chemotherapy drugs were too strong to be given systemically. In other words, if they injected the amount that they needed to kill the tumors into the body, it would also kill a lot of the body cells. And so they just put them on a shelf and said, these are great drugs. They work really well, but they're too toxic. So with the antibody-drug conjugate, you're actually attaching the chemotherapy agent directly to the cell and injecting it into the cell. And so you don't need as much of it. You just need a few molecules of it. And so a lot of these drugs now that were shelved previously are being used in antibody-drug conjugates.
Mm-hmm.
So I'm just going to do a little presentation on a new one that's really exciting. It's a agnostic drug approval. I'm just going to open up a PowerPoint here.
And I just want to say, and just having a look at this information that you just sent me prior to us hopping on this call, just to define an agnostic treatment. A tumor agnostic treatment is a drug treatment that is used to treat any kind of cancer, regardless of where in the body it started or the type of tissue from which it developed. And this is really important because in standard of care diagnostics, so often they're still identifying the type of cancer and the treatment for the patient based on it's in the colon, it's in the breast, it's in the prostate. And they're still so kind of stuck in that zoomed in lens specific to where a primary tumor. These drugs, it doesn't matter. We don't need to know with confidence the origin of your cancer, do we?
Yeah, and that's, I think you hit the nail on the head perfectly there. Precision oncology is what we call rational drug design. You have the target, you have the molecular features, here's a drug for that. Yeah. Versus a lot of guesswork when you're doing the statistical method of traditional oncology. Yeah. You have a breast tumor, and traditionally breast tumors with your receptors will respond to this and this and this. Yeah. And too many people slip between the cracks. And this is where precision oncology really is excelling.
Mm-hmm. Yeah. And the use of the precision oncology specifically, in my brain being starting with enhanced imaging, with thorough genetic testing, not just small panels, that personalized research, and just making sure you understand the molecular feature of this person's cancer. And now we get to these awesome drugs. Mm-hmm. I just want to say a couple more things too, just for folks who are listening. Sometimes as soon as people hear the word chemo, it's like, no, I'm out of here. Not for me. We've heard so many horror stories. Sometimes people just have it in their mind. If I ever get cancer, I'm not taking chemo. And you just kind of talked about the toxicity of certain chemotherapies, including these drugs. But what's specific about this, I'm sure you're going to go into this in great detail, but this is just for those of you who are about to click off, because chemo, I want you to understand what Alex was saying here. When we think about standard chemotherapy, we think of it as kind of a shotgun. It goes into your body and it kills all rapidly reproducing cells. And the hope is that it's going to kill more cancer cells than the healthy ones and that you'll still be around after the chemo. So of course it has strong side effects. And of course people are not interested in that unless they absolutely have to. This is a very different thing. Mm-hmm. What we're talking about here is essentially a targeted chemotherapy. Yeah. Right? Again, I'm not trying to steal your thunder, but just want you to understand we're talking about an incredibly small amount of chemotherapy that goes and directly attaches to your cancer cells. Mm-hmm. So it's a very different thing. The side effects and the outcomes are very different with this type of therapy than they are with standard chemo. I just want you to know that, so that if you have it in your brain, no, chemo's not for me. This is a very different animal, and it's definitely worth your attention in terms of the results it's providing to people and the minimal side effects in contrast to standard chemo. Right. That's all I wanted to say.
Yeah. And you hit the nail on the head there too. Cancer treatment, the problem has always been isolating the cancer cells and targeting them specifically. So it's the problem has always been a drug delivery problem. We've had drugs that can kill tumors for a long time. We just don't know how to have them go directly to the tumors without killing normal cells. Yeah. And this is what ADCs do. They've solved this problem. Wow. So I'm going to talk about a really exciting agnostic drug approval. I'm going to move on here. And as you mentioned, agnostic drug approvals are for a drug based on a mechanism or a molecular marker. And so the next slide that I borrowed basically talks about some of the timelines of some of the approvals. So NTRK fusions.
Can I ask you, are you sharing your screen, Alex? Are you showing us your presentation yet?
Oh, sorry. No, I don't think I am.
I'm eager to hear. Apologies. But yeah, show us what you got there.
Okay, off to a good start, huh? Mm-hmm. See how excited I am? I totally forgot to share my screen. Okay, agnostic drug approvals.
All right.
Here's a timeline of some of the drugs that have been approved for agnostic usage. NTRK1 is a gene that can be altered in fusions and mutations, and a class of drugs called TRK inhibitors. Pan-TRK inhibitors came out in about 2014 or so. And as you can see, there's a couple of different mechanisms defined by NTRK fusions. And then larotrectinib one of these drugs that targets NTRK fusions and entrectinib have both been approved. So 2018, larotrectinib was FDA approved for any tumor that had an NTRK fusion. So that was one of the first diagnostics. And then, of course, we have pembrolizumab. Believe it or not, 2011, the immune therapy PD-1 inhibitor. So it's been around quite a while. We first started using it around 2014, prior to that, actually, after the keynote studies. It's immune therapy, but they found that it worked really well on microsatellite unstable tumors and tumors that have dysfunctional mismatch repair deficient. It's called mismatch repair deficiency. So DMMR is mismatch repair. It's a type of DNA repair that repairs damage to DNA. And when it doesn't do it properly, you end up with mutations. Microsatellite instability. Microsatellites are little repeats of DNA that are located throughout the body or throughout the genome, sorry. And the location and the amount of those repeats is unique to every person. So when you have a high or mismatch instability high tumor, that means that there's a lot of variation in those mismatch in those microsatellite areas. So the tumor looks very different and is more easily recognized by the immune system. And then we also have the T mutation burden high, which, as you know, I really pushed Foundation One to include in their early days. And that means if you have more than 10 mutations per million base pairs of DNA, then you get an approval for, in solid tumors this is, and you get approval for pembrolizumab. Now, we know a bit more about that now. We know that some cases of high tumor mutation burden, pembrolizumab is not that effective, but in many cases it is. And some of the long-term durable responses we've seen with this drug have been in patients that have had very, very high tumor mutation burden, such as ovarian cancers or endometrial cancers and a low-grade neuroendocrine that we had an exceptional response with. So this is just the approvals of other agnostic drugs.
These are just, for example.
Yeah.
Okay.
Yes. So in August, oops, sorry. I'm just trying to move this out of the way.
I can't handle the spelling mistake there. It's going to drive me bananas.
Okay.
Mm-hmm.
August. Okay, that means August.
Yeah, okay.
It's thrown together.
All right.
2023, the FDA granted a breakthrough therapy designation for a drug called trastuzumab deruxtecan. And what trastuzumab deruxtecan is it's a ADC that has been well established. It's also known as TDXD, brand name Enhertu. Basically, what this is it's the antibody trastuzumab. Also known as Receptor. It binds the HER2. It binds a certain region of the HER2. There's also pertuzumab. Pertuzumab binds a different region of the external portion of the HER2 receptor than trastuzumab. But trastuzumab will bind the receptor. And you can see on the end of trastuzumab, it says MAB. That stands for monoclonal antibody. And any drug that has two names like this is typically an ADC if it has MAB in the first name. So trastuzumab refers to the antibody that binds the HER2 receptor, and deruxtecan refers to the chemotherapy drug that gets injected into those cells that is attached to this. Okay. So if you have a HER2 positive solid tumor, including metastatic colorectal cancer, then this drug has this cross-tumor designation now. And so the actual designation is for the treatment of patients with unresectable or metastatic HER2 positive solid tumors that have progressed after prior treatments and who have no satisfactory alternative treatment options. So if you have HER2 positive cancer and it's a solid tumor, then this drug could be a real game changer for you. So I'm just going to go through a few more slides. Okay. So what is the HER2 receptor? So it's a cell surface receptor. It goes both inside and outside of the cell, and it acts as a docking station for a ligand. A ligand is something that binds onto a receptor and turns it on the cell, and then it creates a molecular program. So a ligand, any type of molecule, binds to the receptor. When it binds, it is triggered to change shape or to initiate some type of cellular response. In the case of the HER2 gene, that would be causing reproduction of the cell. And so if you have a lot of HER2 receptors then the cell's constantly reproducing and growing. And obviously, that's what cancer is. So once the ligands have docked the HER2 receptor, this sets off a signaling cascade that goes all the way to the nucleus of the cell and creates a change in the cell. In this case, it's growth. So how do you test for the HER2 receptor? There's two ways of doing it. Immunohistochemistry. There's actually three ways, but the main ways that were used in this approval, and that's the important thing, is immunohistochemistry. And that's where they use an antibody that stains the HER2 receptor a different color. So they'll have an antibody for the HER2 receptor attached to a dye, and then they'll just look at the tumor, and then they'll spread the antibody all over it with the dye, and whatever cells light up with the dye, they know have that, and then they count the amount of receptors in the cells that are positive for it. So IHC scores are typically based from 1 to 3 on the ratio of HER2 positive cells, and 1 is considered negative, 2 is considered, in between, still low, and then 3 is considered positive. And typically, if you do get IHC testing and you do have a 3, then often what they'll do is they'll go ahead and do something called FISH or ISH, fluorescent in situ hybridization. Basically, what this is kind of called chromosome painting, and it just looks for copies of the HER2 gene. So if you have more HER2 receptors, then potentially you have multiple copies of that gene too. And that's a common mechanism in cancers. Cancers often copy oncogenes, and HER2 is considered an oncogene in this case, and they'll produce many copies of it. And so not always are those copies functional. So we've seen multiple copies of a gene that didn't result in high expression. But if a gene is copied in a functional manner, then when it does get turned on, and if there's multiple copies, then you're going to get much more of that gene product. In this case, it's HER2. So actually what the studies show is that if you do have HER2 positivity by IHC, and you also have it positive by FISH, then you get a really good response from this drug. So it's a double impact. And so, usually these tests are done. The important thing is this approval was based on something called IHC 2+, and I'm going to discuss that for a minute. So traditionally an IHC score of 1+ and 2+ are considered negative, and a 2+ is considered equivocal. For the purpose of Herceptin and pertuzumab and other HER2 positive drugs, often a 2+ score is ignored. Not enough of the tumor contains these receptors, and therefore, using the drug is only going to kill a small portion of the tumor. However, many ADCs, and I believe T-DXd was one of the first ones, have something called a bystander effect. And the bystander effect is when they attach to a HER2 positive cell in a tumor, they release a chemotherapy. And then if the neighboring cells don't have HER2, what happens is the chemotherapy agent actually passes from cell to cell, and it kills the ones that don't have HER2. So because of this bystander effect, this drug actually even works in IHC 2+, which is considered low, and IHC 1+. I've seen responses in patients that have only had an IHC 1+ and had a really great response to this drug. So this opens up the designation for many people that were normally considered HER2 negative, 1+ and 2+ scores. This drug can now work for them. And so I'm going to show you how the bystander effect works. So as you can see on the top, you see that little kind of round Y-shaped molecule. That's an antibody. Antibodies that traditionally look like Ys in all text for biology. And then you have the HER2 receptor, which is those two blue circles connected by the line. And you see the internal portion and the external portion. So what happens is T-DXd, trastuzumab deruxtecan, binds the HER2 receptor. That little yellow thing is the chemotherapy agent, deruxtecan. And what happens is it binds to the cell. The cell takes in the receptor and the antibody and the chemo. The chemo goes into the cell, kills it from the inside out, and then you can see it releases it through a couple of different processes. Basically diffusion through the membrane, which is right up here, as you can see in the middle slide. And then at the bottom, you see phagocytes. They can also release the chemotherapy drug into the neighboring cells. And so this is how this bystander effect works. So just to summarize, because ADCs, antibody-drug conjugates like TDXD, target unique receptors or cell antigens, they can be used for any cancer that expresses HER2, literally. And so now we are testing by IHC every single cancer that comes into our lab. It's just something we do now. I wish all medical facilities did that, but it's not that expensive. I think it's about $1,000 or something to get a HER2 test from a approved laboratory. We test everyone. Why not? Because if this drug is going to be a benefit for them, great.
It's really
so additionally, the bystander effect of TDXD allows it to work in HER2 low or even tumors that don't have HER2 receptors on every tumor cell. And that's the bystander effect. And TDXD is only one of the many ADCs in development. I mean, we literally see a new ADC every 2 weeks, and we constantly upgrading our list of what we look for. So how do you test for these? Well, one of the ways of testing for these, and this is something that we do a lot these days, is RNA expression testing. So you can tell, yes, you can do pathology for each individual receptor, but the problem is if you do it that way, you're going to be using up your tumor sample before you even get through maybe 5 or 6 different tests. So we have a solution for that. We test almost 21,000 genes at one time. We look at RNA expression, and just about every single ADC that has come out is in our panel. And any new ADC that's going to be in development will also be in our panel, because we're looking at just about all of the genes in the human genome. And so I'll give you an example of what that looks like. So right here in the column B here, you can see this.
Mm-hmm.
This is the gene name. So this is the name of the gene that we're looking at. And then over here is what we call the reads per million from the tumor. And basically reads per million is a way of determining how many RNA molecules you have for a specific gene. And so out of 1 million molecules of RNA, this one here, AKT3, had 2,062 molecules. And so in the tumor, that is. And so then what we do is we compare the amount we find in a tumor to a normal cell, because every cell is going to have a different level. So if you notice here in normal peritoneal tissue, the amount of this AKT3 normally there is 283.88 molecules per million molecules of RNA, whereas the tumor has 2,062. And that gives us a fold change of 7.2. So it's highly overexpressed. If we look in normal liver, you can see the normal level is only 39. And so that gives us a fold change of 52 compared to normal liver. And then we look at different lymph nodes, for example. This lymph node, it's normally they're present at 344 compared to 2,062. So if we look at across all of the tissues in the body, we can see this AKT3 is highly overexpressed. And therefore, any drug that is going to be targeting that AKT3 is going to be very effective in this person's cancer across all of their metastatic sites. So this is our short list that we're using right now of antibody-drug conjugates and different genes that are targetable and so on. So for example, this one here, this is an antibody-drug conjugate. Now, unfortunately, in this patient, there's no expression of it. So that antibody-drug conjugate would be useless in this patient. And then there's another one here, CDH6, that we just added. A new conjugate, new drug that just came out that targets CDH6. It's in clinical trials right now, and it seems to be working quite well. And then here's another one, the sodium receptor. As you can see in this patient, the sodium receptor in the tumors is 14.9. And then in their normal peritoneum, it's 11.04. So that's only slightly overexpressed. So we wouldn't expect to see a massive benefit from drugs that target the sodium receptor in this particular patient's peritoneal. However, in the liver, which normally has a much lower level, then the relative level the relative fold change is 100. So we'd probably see a benefit of this drug in the liver, but not in the peritoneum. because it's relatively not overexpressed. Now, here's one here for the mesothelin receptor. There's a new drug that targets this. And as you can see, it's barely expressed in any of these normal tissues. Maybe one in the normal lymph node and, 0.2 in this lymph node. And, it's 0.4, whereas in the tumor, it's 11.9. And so that's a 24-fold overexpression compared to the peritoneum. That is a 11.92-fold overexpression compared to no expression in the liver. And then this is a 6-fold overexpression compared to normal lymph node, and so on and so on and so on. So we know that this drug that targets MSLN is going to be relatively expressed higher in the tumor than all other normal body cells. And therefore, that drug will be beneficial for that patient. So whenever a new one comes out, a new ADC comes out, we add it to this list. And see if it's going to benefit that person. So I'm just going to go back to our presentation. Why don't you explain this slide on how you could get this RNA expression?
Oh, yeah.
Testing?
Well, let's go back over what you just said first, okay? Which is essentially, instead of, what has been the case for cancer patients for pretty much ever, until the super recently, which is the diagnosis being based on what the, surgeon, radiation specialist, medical oncologist.
Tumor location.
Yeah, right. Based on what they think that kind of the primary location of the tumor is. And then, okay, let's try this chemo, or let's try this drug, or let's try this drug and see how it works for you. With precision cancer medicine, as a cancer patient, you don't have to be the guinea pig anymore. You can find out before you try a treatment how likely it is to work for you. And Alex just kind of showed you this chart. And that's just one type of testing. You almost certainly have heard of DNA testing. He also talked about the HER2 IHC and things, but you can identify through a tumor biopsy or surgical remnant that will be around. Hospitals hold on to these things for literally decades. You can identify throughout your entire genome which of those genes are a problem relevant to your cancer, and to what extent. You saw the ratios there. So which are the most relevant or most prevalent mutations or molecular features of your cancer, and which drugs have been designed to target those. So you don't have to try 3, 5, 10 different drugs and wait for however many months on the CT scan to see if there's any change. You can know before you start the drug the likelihood that this drug is going to be suitable to you. Yeah. And there are so many of these drugs, as Alex is saying. And that's what's exciting. The other thing that really excites me about these new antibody-drug conjugate drugs is how little of the chemotherapy, how little of the drug is required in your body to be effective. And again, as I started out, as Alex was talking today, that's a very important piece. We want your quality of life during and after treatment to be as supreme as possible. And of course, the less side effects you have during treatment, the better your quality of life, faster recovery, and all of those things. If we can minimize those common side effects, that's ideal, and directly targeting things. So this RNA test that Alex is talking about here, there's a way for you to find out a little bit more about it. We made a YouTube video a few months ago, and I've linked that here. This will also be in the comments section too, to make it easier for you to access. That's where Alex talks more about how that test works, and a variety of different targeted therapies that you can be matched to. I've also added a link here in case you want to talk to Alex about your case specifically, so that you can just click there and book a meeting with him directly. This consultation, we gather all of your medical records. Alex and his team do a deep dive into everything that's known about you, but also relevant possible treatments. There's a big difference, again, between standard of care, and patients having to kind of go through a certain menu and a certain order of treatments before perhaps even genetic testing would be suggested for them, or any sort of these newer types of therapies might be recommended. Our job, obviously, is to get you on the right treatment right now without you having to go through all those hoops and, again, kind of essentially be a guinea pig or have guesses being made when you're dealing with a life-threatening illness.
Yeah. And a perfect example is, when we do this testing, this RNA testing for patients, we have this massive Excel file. The patients sometimes don't have cancer. They just want to have a plan for the future. And then we'll get this call and, I'll get a call from the patient, maybe a year or two later or something. And they'll say, oh, my cancer's come back. My doctor wants me to go on this new clinical trial. It's a new drug. And I'll say, okay, why does the doctor think that this is going to be good for you? They said, well, he's not sure, but, they think it's going to be good for me. I'll say, okay, give you the name of the trial. And so I'll take the name of the trial. I'll look up the drug. I'll find out the mechanism that it uses, what it targets. And, for example, a case was a patient called me up about this drug that targets something called LIV2, or I'd never heard of that receptor, or sorry, LIV1, I believe it was. I'd never heard of that receptor or the gene or anything, but apparently there's a drug, and I looked into it, and it's an ADC, an antibody-drug conjugate that targets LIV1 receptors. So I just opened up the patient's clinical data. I looked at their Excel file and found that they'd had really high expression of this LIV1. And so I was able to get back to them within about 20 minutes of talking to them and say, guess what? I think this trial is going to be a real game changer for you. I had the same case, the same type of cancer, and the patient asked me about the exact same trial. And I looked up on their form, and they had very low expression of LIV1. And so, unfortunately, I had to say to the patient, well, I don't think LIV1 is going to be the best for you. But while I was going through your file, I noticed that you have this MH6, high expression of MH6, and there's just a new drug out for it right now. And it's in a solid tumor trial. So that's the trial you should go on. So it really does allow us to figure out, what to do.
Yeah, really to zero in very quickly. So I guess another point you're making inherently in that is that you don't have to redo this testing.
No, you do it once and that's it.
Then we know what's going on. And then, if it's 6 months or 2 years down the road and some new therapy is being recommended, it's really easy to have confidence that it's going to be beneficial or not for you. And of course, as a cancer patient, making sure you're on the right drug, that you're not wasting time and you're not experiencing unnecessary side effects is just what it's all about. So this is an excellent way, if not the best way right now.
It is. Yeah, it's one of the greatest combinations of testing and drug development that have just met in the middle. And, they both happen at the same time. And it's just been incredible for cancer patients.
A point that I also like to add when you're talking about the how many of these new therapies are coming out, whether it's daily or weekly, but, so often what we've seen, because we've been doing this for 14 years now, so we've been been there, done that, seen it all kind of thing, is that even if the best option that can be identified for you right now is one that's going, say currently your doctor is saying, well, 6 months or whatever, and we found an option that's going to say give you 3 years, that still better than 6 months, but obviously not the ideal you're looking for, which is complete remission. But numerous times we've had the experience where just helping someone to have that much extra time has allowed science in that window to develop new drugs to give this person even more time.
Yeah. So our goal as a company, as you know, is to turn cancer from a deadly disease into a chronic disease. We're not at the point where we can cure it in most cases, when it's a late stage cancer. But if we can control it and allow that person to live a healthy life and not die from their cancer, that's a wonderful goal. And I believe we are getting there now.
Yeah,
I believe we're very close to that, with testing and these new ADCs, that's a huge piece of that.
Yeah, absolutely. And so much data that can be utilized to your benefit. So enough said for today, I think. Thanks, Alex, very much for joining me to talk about this.
Yes.
And if you'd like to find out a little bit more, links for how to contact us will be in the comments section.